So, it's the reason why the question was perfect for you. Now you just joined us, so Poino, you're Senior Vice President of Alnilamo. You work on research department, and then you developed this RNA interference product, which is one of the first ever approved. So can you tell us a bit more about your strategy and what's the difference between all the others that were presented here?
Yes. Well, thank you for the introduction, and I'm happy to say I think siRNA is only getting started, and you're going to see a lot more from not just Alnylam but a number of other companies in the coming years. So, you know, as you mentioned, we had the first approval for an siRNA medicine in 2018, which was an LNP containing an siRNA to knock down a gene called transthyretin for the treatment of hATTR amyloidosis. And since then, we've had a subsequent 4 other medicines approved, but all of these are chemically modified siRNAs with a conjugate, which is called GalNAc, that's on one end of it, that allow the siRNA to be targeted to the liver. And it's sometimes I have to pinch myself because it's like science fiction. It's some of these medicines you can give to patients once every 6 months because they're so long-lived in the cells of the liver. You have this very, very long duration of action, and it takes away this challenge with compliance where a patient, you know, needs to get up in the morning and remember to take their tablet. They just get an injection once every 6 months, and they have this clamped pharmacology. So it really is an amazing platform. To answer your question more directly about our strategy, you know, I mentioned that we can target liver. So of course, any gene in the liver that either causes a disease or we think if we remove it, it will protect people from a disease are perfect targets for Alnylam. And we've moved more in recent years from very much focusing on rare disease to more common conditions. And now we've also gone into neurological diseases. So we're in the clinic with our first CNS program with a different conjugate to try and develop a treatment for Alzheimer's disease. So wherever we can deliver siRNA to any tissue, there are targets that we can pursue, and we think will be great of great therapeutic benefit to patients. The challenge is being able to deliver, and we can do it to the liver, we can do it to the brain, but we need to have ways of delivering these medicines to other tissues to really unlock the full potential of that type of medicine.
Yes, because now we work on cardiometabolic diseases, infectious diseases, central nervous system, ocular. But there are other fields you'd like to address. You can do.
Yeah, there's a number of other fields. I mean, you could, unlike a small molecule, which you would take orally and it's distributed all around the body, an siRNA is not like that. It has to be targeted to particular cell types. So, for example, if we want to treat a disease of the kidney, for example, we need to have a way of driving the siRNA into the right cells in the kidney.
What's your strategy for
so the strategy for us is finding solutions to delivery. We are not limited to therapeutic area. That's not the way that we think about what we do. We think about siRNA as a platform. If you can unlock delivery to a tissue, we will address any disease in that tissue. And you only need to look at what we've done in liver. We have a treatment for peripheral neuropathy, cardiomyopathy, hopefully soon, a kidney disease called PH1, a disease called porphyria, which is sort of unspecified pain, hypercholesterolemia. But this is all because that's the biology of the liver. These are the things it's involved in. So we've been very broad in terms of the diseases that we've treated.
All right. Okay, we'll go back to you. Another approach, we've talked a little about it during the small talks. It's you, Hélène Tran. You're head of oligonucleotide therapeutics program R&D at Servier. So you focus on these very small RNAs, namely as brain therapy, oligonucleotides, and it's very specific in comparison with wider and larger ones that we talked about. So can you tell us more about what you're doing?
Sure.
Yes, it's working normally. Yes.
Yeah. So thank you very much first. And so Servier is focusing mainly on antisense oligonucleotide for brain disorders for the moment, because as opposed to siRNA, the challenge is likely not the delivery, except if obviously you want to cross a blood-brain barrier. But once you're in it, you do get into the cells. And our focus is really to target first all the monogenic disease where we do know the genetic cause, where we do know we can actually impact the disease progression using this small molecule. And again, because we do know the target, we do know the sequence, we can design an antisense oligonucleotide that will have an impact on the expression of the target. So that's, for the moment, our strategy.
So you developed this strategy and you've got a platform, 50 to 100 persons working on it with you, that's it?
Yeah, so we have like a growing team, so it's working like a matrix, so they are not full-time on it, obviously, but we start to get more and more experience in the field. And as you mentioned, we like to work as a platform, meaning that we learn from one project to the other, we learn from one antisense to the other, from one sequence that is not working, not active, toxic, we actually leverage the knowledge that we gather around it so that we avoid to reproduce the same mistake, if you allow me, so that at the end, hopefully, after a few projects, we would be able to go so much faster, but fast in a good quality. And I insist also on it, meaning that we wouldn't work on sequences that we would have shown before that the sequence wouldn't be active or would be not tolerated, acute or long-term. Because again, there was a question about the half-life of antisense. Antisense have a quite long half-life, and the main issue is actually to make sure that we can anticipate potentially delayed toxicities that will appear in the body because of this long half-life.
So your strategy, the advantage is that you can target anything you want on paper.
On paper, yes, because again, it is like targeting RNA, and there is no undruggable RNA. We can target small, long, nuclear, cytoplasmic.
Okay.
Technically, any RNA, as long as it is expressed in the target tissue, and as long, obviously, that it has a role in the disease biology.
So what's very interesting is that you can treat one sole person, one sole sick person. For example, there was this Milasen treatment, which was developed just for one little girl who was sick. It was nucleotide, and it's the perfect personalized medicine dream.
Yeah, so the power of this type of technology, again, because you work as a platform, and I think it would be the same for siRNA and hopefully soon also for messenger RNA, meaning that if you know how to, the process and how to design and how to perform the best screen, you would be able to design quite rapidly one therapy for one mutation and in some cases one patient. And that was the case for the story of Milasen. And the idea here is not to, is really to show the power of the technology. That's my only goal here. Is not to say that we should all go there. It's just to show the power of the technology for super mega rare disease, where there is a urgent medical need, where there is no business model. This type of technology can, because you can go fast, because you would know how to design and to work on it, can yield an enormous potential for this type of disease. That's the only goal here. It's because you know the target and you know how to design your molecule, you can virtually design and develop any molecule for ultra-rare as much as more common disease. Okay,
so we'll talk about it maybe if you want to react to that later. But Annette Sommer, so you are Emerging Science Innovation Lead Oncology Europe at Pfizer. And then you focus on cancer and RNA for cancer to struggle against cancer. And of course, the immuno-oncology is a very important field. How do you address it through RNA, mRNA?
Yeah, perhaps I'll start with going a little bit which now sounds already like history.
Yep.
So like coming from the infectious diseases, coming from COVID-19 and the success, and being very glad to have contributed one of the 2 approved vaccines for COVID-19, which came from the collaboration with the German company BioNTech. And in the past, we had also worked on influenza, on flu. So clearly, we come from the infectious disease angle. Yeah, so this is what brought us into messenger RNA LNPs. And so this is where we are continue to be active, be it COVID, addressing new variants, then flu, perhaps in the future, combine the 2 in one lipid nanoparticle, and then extending to other infectious diseases, for example, respiratory syncytial virus, and so on. So that's really the background, yeah. But now we think it's such a powerful technology. It's perhaps not at the end of it does not yet has reached a plateau of technology development, but as such, it's very powerful. So now we are exploring what else one could do with messenger RNA LNPs. And of course, on the one hand, we are thinking about oncology, but also inflammation immunology. And what really is of interest to us is to look at the CAR-T approaches. You know, the classical CAR-T approaches are based on ex vivo manufacturing. So you remove the T cells from the patient, you transfect them with the vectors, you expand, you do the QC. It all takes a lot of time. You have to have highly trained personnel to do this. It's a lot of logistics and costs. And now asking, what if, and we've heard this and seen it on some of the slides before, what if we could make the human body, the human cells, make the manufacturing themselves and do QC? Of course, we know then we cannot control the QC as well as we can do when we do ex vivo manufacturing. But we see many advantages in this, and it's not only on the manufacturing side, it's also this other topic that we've heard this morning by this, like handling these tricky cells, the T cells. Some of them lose functionality, they may even differentiate too much and so on, and then in the end lose the power to really have a beneficial effect on cancer. Yeah, so that's where we come from. So the idea is then, can we do CAR-T by in situ immunotherapy? Yeah. And so I think this is really a fascinating topic. And yes, my title is Emerging Science and Innovation. So I'm looking for those topics where perhaps the solutions are not yet ready or are only about to be developed and then to be brought into the clinic. And there are academic teams, biotech startups working on this. And then delivery can either be then what we heard in the morning, for example, lentiviral delivery, and then using DNA, but also using messenger RNA, LNP, expressing the CAR. And yeah, so I think these are very interesting opportunities. And I think it goes beyond. It does not have to be T cells only. It can be NK cells. I think on the slides by Nathalie, we've seen the NK. I've seen the Chantal. Sorry. We have seen NK CAR as one example. Yeah, it could be myeloid cell macrophages. And another topic I think we have not yet discussed is to go beyond the CAR approach. And for me, what is really an upcoming topic is really to look at transcription factors. Transcription factors that characterize a certain cell state, be it the immunosuppressed or the immune activated. And so what could we do? Could we use messenger RNA LNPs to bring the right transcription factor into the system and turn the switch to the side that we need? And now be it oncology or be it very often the opposite in autoimmune disease. And we all know, and perhaps I like that you said, Paul, you go for the liver, because this is where your technology works best. And from this, you then address different indications. And I think we've heard this many times today, but what we all have to work on in academia, in biotech, in pharma is targeting. So it's still the delivery, and this is really the call to work together on improving delivery. There are startups, academic groups that work on what I call passive de-enrichment of the liver solely by changing the composition of the LNPs, the right mixture, using AI and mouse-based screening and so on, but then also using targeting, and be it aptamers or SCFVs or antibody fragments. So I think this is key for me.
Okay, thank you very much. So Chantal, delivery and targeting are the main issues today? Maybe toxicity too?
Maybe toxicity too.
Yes, it's one.
Yeah, as I said in the challenges, but yeah, you're right, Annette. I mean, we need to find out how to target. I didn't have time to say that there are a different, you can actually add your ligand on your LNP in a different way, and you can use a very easy way that actually you can see in the literature, just using this by Malay-made or something like that conjugation. But then we have to think, what will be actually such LNP with those different types of ligand using this specific conjugation once it's inside the body? Will it trigger another types of side effect due to another types of reaction, like retromycal reaction, which can be very toxic for the body? So I think yes, there are. I would say modifying, detargeting by adding a specific component to go in a specific way. I think it's already, in my opinion, you have to do this first, but you need maybe to have another. Maybe you have to find out 2 levels of targeting, not just the one with targeting. Yeah, this is. I don't know if you agree with me, but this is.
Okay, thank you. And Pierre, well, we've already talked too, but there are many issues. What about your mRNAs, which are larger than those that we talked about, nucleotides, which are sometimes difficult to engineer and to deliver again? Is there a problem of, for example, immunogenicity? How do you address these problems?
Well, we have decided to use this mRNA platform because, first of all, it has been validated through COVID, for example. And we think the fact that you can express a protein gives you a huge number of applications. You can have intracellular proteins, extracellular proteins. You can, of course, create or recreate a function which has been suppressed in rare disease, and you can modulate immune expression, and you can also, of course, inhibit other proteins. So we see this as a very broad platform. That's why we have chosen it. As a platform, as you said, that's something that should work for multiple mRNAs. And it has value for vaccines and therapeutic applications. This being said, I think there will be space for multiple platforms. I mean, there will be space for siRNA, small RNA platform for sure. Maybe not within the same company for obvious priority and financial reasons, but across the industry, I'm sure there will be space for multiple platforms, and nobody knows who will be the winner at the end, right?
So you talked about your mRNA manufacturing in Neuville-sur-Saône. Mm-hmm. Okay, so when will it be ready?
Good question. It should be ready within 2 years for commercial manufacturing now. Okay. If everything goes right.
4 vaccines?
4 vaccines? You mean? Yeah.
To get 4 targets, 4 different vaccines. That's right.
No. It will be ready, and I cannot tell you today how many vaccines will be ready at that time for commercial manufacturing.
You would like to perform 4 in the same time. In the meantime.
That's what you have seen on the slide. Yeah, no, that's the facility has the ability to manufacture 4 vaccines in parallel. Whether it will be the case in 2 years, I cannot tell you, but that's the way the facility was designed to be able to accommodate 4 modalities at the same time.
So we could understand that you will not focus on COVID, of course, after what happened. Yeah. You were a bit late, we can say, on that point. Absolutely. So what are your targets?
Well, our targets are respiratory disease that I have seen, flu, respiratory syncytial virus. We are now expanding to bacterial disease. I have mentioned acne, chlamydia as our future targets, which are getting prepared for clinical trials now. And then we are expanding to other therapeutic areas beyond vaccines, which are, as mentioned by Annette, oncology, rare diseases, and immunoinflammation.
So you deal with nearly every field you can that could be addressed by mRNA. Okay, very interesting. So there are a few issues, general issues. Regulatory authorities, regulation is something very important to point because you can be very quick when you find something. We could see it with COVID. There had to be a very quick reaction. And so there's maybe you worked in the US too, and you could work very easily with regulation. You could have some product developed quickly. And in France, of course, it's a bit more difficult.
So when I was in the US, it was a different frame, meaning that I was working also in an academic lab, and we were working for one patient. So in that context, the relationship with the regulators is very different from a context in the industry for many more patients. So I'm not sure that the difference will rely solely on the geography, but it will also be the context of who's discussing with the regulator. And I'm not sure that I am in the right position to say whether Europe versus US, there is such a difference in terms of regulatory response, reply, agility. We are seeing that they are actually supporting us, European as much as US. But as someone alluded before, different mindset and different questions and different backgrounds also.
Okay.
Yeah, and
yes.
Maybe I can just add to that from our perspective at Alnylam. Actually, developing our therapies in Europe has been a key part of why we've gotten to where we are today. It's I think someone mentioned in an earlier panel, you know, if you take the MHRA in the UK as an example, they take a much more pragmatic approach to novel therapies, and they're willing to work with you to bring something to patients. The FDA tend to be more challenging to work with on these novel therapies, to be perfectly honest. And our first trial that I mentioned, our LNP and patisiran, we had a key center in the UK that we developed. We had a key center here in France, actually, with Professor David Adams working in hATTR amyloidosis, where we were able to run the trial. So I think there's.
So France was quicker than the US?
Yeah, I think there's a lot to be said for the. So if you want to critique Europe in any way, you could say the earlier stage research and bringing the novel therapies forward lags behind the US. But I think actually in terms of what you can do in the clinic and getting things to patients, Europe is actually more forward thinking.
All right. Annette, is it your experience?
Well, I just attended 10 days ago a meeting in Berlin, third messenger RNA-based therapeutic summit, and they had invited also speakers from Swissmedic as one unique country, Switzerland, and then from EMA. And really the message that they tried to convey, and regulatory specialist from Moderna was leading the discussions and through this session, so was really trying to support even very complicated situations on the molecular side, So be it LNPs with more than one messenger RNA in it, which is new, And so I see them more on the supportive side, and of course they say they have to apply their the scrutiny to look at the quality control and all these aspects. But that's what we want too, because that's for the benefit of the patients. So I rather see the role of these the regulatory authorities on the I'm talking now about EMA and Swissmedic in this case, very much on the positive side.
All right. Okay. Thank you. Do you have first questions? Any question at that time? Yes. Mark.
Yes, I was wondering to what extent, well, you mentioned this exciting possibility to generate CAR T cells in vivo. So I was wondering how mature was this approach, whether the feasibility has been demonstrated in preclinical models, and what kind of challenge you can contemplate based on the analysis of these in vivo preclinical studies.
Yeah, as I mentioned, thank you for this question. So as I mentioned, this field is strongly driven by a number of early-stage biotech companies. So there are examples, and one is, for example, Interius in the US that work on lentiviral delivery. So then DNA is then the vector. And then company like Capstan Therapeutics using messenger RNA LNV plus. Not everything is published because it's early stage biotech on using messenger RNP in a targeted version for CAR-T. Yeah, so that's early stage. Many things are not published. Yeah, so and things are not yet in the clinic, but I think that's a field that we are very actively following. Yeah, and that's what I mentioned. I'm the one going for the areas that are not yet fully mature. Yeah. Mm. Yeah, but that I find really exciting, and that may be game changers in the future.
Yeah. And there's a company called Tidal Therapeutics that does in vivo delivery of LNPs. This company was acquired by Sanofi a few years ago.
That's
Stay tuned.
I did not mention it.
All right. Another question? Yeah.
Yes, about this problem of in vivo delivery, the question is the specificity. Of course, when you inject, for instance, LNP, at the site of injection, you have a very high concentration, and then it's going to get diluted within the body. And we know from in vitro data with any kind of delivery, whether it's LNP or viruses, that the concentration matters a lot. So what's your opinion about that? Do you think it would be one day possible to target any cell type on wheel without any trouble of lack of specificity?
Okay, who wants to answer?
Well, I can make a couple of comments because I mean, I reflecting on our experience with LNPs, our first medicine is actually given intravenously, so you don't have it's not accumulating in the subcutaneous space, for example, and that targets the liver because it binds to ApoE and it's taken up by the LDL receptor. Our other therapies, however, don't employ LNPs or viruses. They're what we call conjugates. So these are siRNA molecules where we've chemically modified the nucleotides so they resist nuclease degradation. And then we add a targeting ligand onto one end. And that is very specific to a receptor that's expressed on the surface of hepatocytes. So it drives the tropism in that direction. So our sort of approach is, yes, you can use an LNP, and maybe you can modify the LNP and insert proteins into the membrane, et cetera, but or use a virus. But for us, finding ligand receptor pairs that are specific for different tissues and cell types within tissues is our approach. So we make these conjugates and we try and drive them towards those cell types with the receptor only being expressed in those cells and adding the ligand onto the siRNA.
Thank you. You want to add something?
Yeah. So, Paul, the question is that if you make the conjugations of your the molecule of your cargo on is it enough in terms of concentration that are actually taken up by the cell, because you have one siRNA plus when you do your conjugate. Do sometimes, maybe depending on, it's really open question, maybe depending on the type of mRNA that you would like to address, target, the amount of the conjugates is not there, or am I wrong?
Yeah, so we we do a lot of studies looking at the accumulation of our medicines in tissue. And so an example would be something like inclisiran, which is medicine we discovered at Alnylam and Novartis subsequently have rights to and commercialize. But that's 25 milligrams once every 6 months, sub-Q dose. So you don't need a lot, and that. Remember, once every 6 months. So what it's really driven by is, yes, you need good uptake by the receptor. You don't need a ton of drug, but you do need a potent siRNA molecule that knocks down the target. So as long as you have something that's very potent, and we spend a lot of time finding the right sequence, then the dose actually can be actually very reasonable.
Yeah, I just wanted to comment on the term targeting. Don't forget that there is passive or natural targeting of LNPs, right? The Alnylam LNPs, if I am correct, when you inject them, they target the liver by their physicochemical design. So this is a very important targeting, natural kind of passive targeting. And as far as vaccines are concerned, there is still a debate on whether you need to target LNPs, because when you inject them intramuscularly, they are naturally targeting the dendritic cells. And some people are saying, well, we should put a ligand on the LNP so they go actively to the right dendritic cells. But some people say, well, we don't need to do that because they naturally go to them. So don't bother. So that's why the term targeting has to be really weighed. What really do you want to achieve, and where do you want to go? And do your LNP already go where they need to go?
I would also like to make a comment to Philip and his question. So I think this type of using AI, combinatorial approaches, computational approaches to work on better or different, very different LNP compositions may allow to do this passive targeting. So first of all, liver de-enrichment, and then perhaps adding certain tissue types to it. But that's the future. We are not there yet. Yeah, we are not there yet. And then the targeting aspect and using just one ligand, or as we now see also upcoming with the antibody-drug conjugates, dual targeting, addressing dual antigens. Perhaps this is something to be explored, but it's still very much in the explorative stage. And I could also foresee that there may be like a desert where for quite some time we are not really making a breakthrough. But I hope we continue to work on it. Yeah.
Great. Yes, another question? Yes, Mark.
Yes, well, what I'm puzzled by the fact, typically, for instance, when we discussed this morning about AAV approaches, so clearly we are in a situation where temporality is not the issue, because basically you administer the vector, it's expressed for a very long time and so on. Clearly here we are in a situation with all these oligonucleotides, either in a situation for an mRNA vaccine where you need just transient expression and then you'll get triggering of the immune system. But for instance, when you want to restore or to inhibit expression of a particular gene causing a disorder, then in this case, temporality and duration of expression is key. So, and this has not been really discussed here. So I was just wondering, what is your feeling about this?
Very interesting. Chantal, do you want to start first, or Paul, whatever?
I mean, if you were, when you give a talk on using mRNA to produce a car, people are always telling you it's not going to be really good because it's transient expression. I mean, I think it's really depending on the type, again, as Paul said. It's going to be dependent on the type of cells, on the type of target that you would like to address, because the Cartesian therapeutic, which is very, I think you are, I don't know, it's really amazing to see that just the Descartes for myasthenia gravis, I mean, their data was really great. They are doing the cells that they are actually modifying and injecting in vivo. It lasts for months. So I think it's, I agree that there is a transient expression, but if you have, if the cells are not so much dividing, maybe you can stand and not forget about the fact that it's not toxic. I think the toxicity really, I mean, does matter because it's not toxic at all.
It's a very good question. It's something we've thought a lot about Alnylam, particularly as we've gone from treating rare disease to more common conditions. And I'll give you a good example. So we have a medicine now that's in phase 2 development to treat hypertension. It targets the liver, it knocks down angiotensinogen in the liver, and you get this beautiful drop in systolic blood pressure that lasts 6 months. You give this medicine once every 6 months. What happens if that patient has an issue, if they have a trauma, if they have to undergo some routine surgery, all the conditions where you would typically stop administering an antihypertensive. So we're actually co-developing what we call it a reverse here. It's basically an antidote to the siRNA that you can give, and it immediately mops up the drug in the liver.
Really?
And it will bind to this instead of to the target, and you restore expression of angiotensinogen and you restore blood pressure. Whether it's used or not, I mean, these patients still respond to pressors, and you can give them salt tablets and their blood pressure will go up. Whether it's used or not, I'm not sure, but certainly in terms of encouraging physicians to prescribe the drug and feel comfortable that they can reverse the effects should they feel the need to, I think is a really important thing. So yes, it is absolutely a very key point.
Very interesting.
So why not making a drug that does not have such a long lasting effect, and then make a re-administration then?
Well,
I mean,
you could, but the challenge is, like, for diseases like hypertension and hypercholesterolemia is another example.
I know, the PSK.
It's the area under the curve. It's your exposure to high blood pressure and people don't remember to go and to take their medicine every day. So it's one of the big advantages is that clamped pharmacology, people, passively having their disease managed. That's the key, and to date we've not seen any safety issues with it. But it is important to have a way of, just like with for example, in bleeding disorders, these types of antidotes exist where you can reverse the effect. It's a similar concept.
Acceptability is okay. Okay.
Very interesting. Thank you. Do you have another question? Yes, please.
Yes, thank you. I have a comment also probably for Paul about the stability. So I do think it is actually very impressive to have a 6-month stability, and I was wondering if it was really due to the chemical modification of the siRNA, and also your insights comparing to ASO. They are not lasting as long, even if they are GalNAc conjugated. So I wanted your insights on really the reasons beyond the stability of the siRNA effect.
Yeah, thanks for the question, and I will let Hélène chime in on the antisense oligos. But there does seem to be a difference, certainly for siRNA. When we first started, so Alnylam started over 20 years ago now, and most of that time was spent trying to figure out this issue. How do we, you give the siRNA, it's immediately degraded. How do you stop that from happening? And so the solution was actually to modify in the sugar molecules in the 2' position with things like 2' methyl or 2' fluoro, and then modify the backbone of the siRNA. So we use something called a phosphorothioate backbone. These are the key sites that are recognized by nucleases. So that was the key piece of data that allowed us to really stabilize those molecules, not just in the circulation, but also once they get into cells. The other thing that's very, very important, and I think answers the other part of where you were going with your question, is what happens to the siRNA when it gets into the cell. In the case of our molecules that are targeting the liver, they get stored in a compartment within the cell, and we published this a couple of years ago in nucleic acid research, where we showed that the siRNA is trafficked into the cell and it's stored in vesicles, essentially, part of the endolysosomal compartment in the cell, and gradually leaks out over the course of months. Oh. And it's that, I think, compartmentalization within the cell, coupled with the chemistry, that means that it lasts for such a long time. And since we've learned that, we've actually been able to go back, and now we're able to design medicines which we think can be given once every 12 months, now that we understand the rules of what's actually happening. So you can push the duration even further just by sort of tinkering with that system. But those are the two big reasons.
This is fascinating, really. Yep.
And basically, this is the same situation for the oligonucleotides that are...
No, we do not have like the same duration of effect as the siRNA. The 6 months duration of effect so far has not been like widely seen for all compounds like Anilam showed. There are some examples of long half-life for antisense for like in a trial for Ionis for tau, for example. But I believe that because the storage within the cells in the liver, I guess we are using the same conjugate, the GalNAc, and the antisense would use the same mechanism. However, the difference, and here I'm talking about gapmer approach, where you have a modified antisense, you have like a heart of DNA and a side of RNA, might actually explain also why perhaps those antisense are more prone to degradation, while for siRNA, fully modified RNA modification, there is something here that definitely needs to be investigated if you want to get to the level of the duration of action here.
That is a very key point, that we can modify the whole siRNA, whereas with an antisense, you can't, because you'll inhibit cleavage of the transcript if you modify the whole thing.
Okay, thank you very much for participating in this panel. Thanks a lot.